Good evening. I'm Naren representing Team 109. Today I will present our project titled Analysis of Plant Cell Shape. We actually selected one of the projects provided by Professor Edgar Loberton in his folder. So our motivation is mainly as this is a research project that is being currently done. We are trying to answer one specific question that was raised during the research. So this project mainly focuses on exploring the variations in the plant cell expansion due to the presence of one specific protein called mutant cellulose synthase. In simple words, what we can say is, does the presence of this protein really affects the way the plant cells expand? So our main objective is to calculate the circularity index for each and every cell present in the plant leaf. Uh, why we are calculating the circularity index means we need to check whether the cell is in a proper polygon shape, or is it getting expanded and becomes a mere circle. And for this reason we are calculating the circularity index, and for this we are building an instant segmentation model which segments each and every cell present in the image, and we map those features that we have to this image to calculate the circularity index. So this is our data set. So we have the images, we have 28 images actually, and we have those rectangular boxes, right? We have around 20 to 50 cells present within those boxes, and for each and every cell present inside the box we have the corresponding values in the csv file. So this specific bounding box have 22 cells in it, and for each and every cell will be having its corresponding area, perimeter, and also the circularity index. So using these two data sets will be segmenting each and every cells from the image, and will be mapping its circularity index to it to calculate the circularity index for a new image that we have. And to assess the performance of a segmentation model, we use the intersection over union metric to measure the accuracy of a predicted mask against the ground truth labels that we have, and to pre-process each and every image that we have. And we can clearly see that the images were not that clear, and also the cell walls that are present inside the image were blurred, and also they were not completely over there. And to map those values and also fill those areas that we have, we enhanced our image quality using the bilateral filtering, which preserves the edges while also denoises it. And we implemented clahe, contrast limited adaptive histogram equalization, which helps us to increase the contrast of our image. And we use dilation to highlight the boundaries that we have for each and every cell. And as we had only 28 images, it will be obviously making our model to get overfit on our data, because it is only a less amount of images. So for that reason we are augmenting our images using albumentation library, which helps us to augment the images along with its corresponding mask that we have. So we used that, and these were few images which we captured right after performing our pre-processing techniques. And we implemented mask rcnn2 as our baseline model. So mask rcnn's architecture generally extends the faster rcnn's architecture by adding just another one layer called segmentation mask predictor layer. So it operates in two stages. At first it has a region proposal network, which identifies the object regions that we have, the region of interest that we have. And the second one does something called as alignment of ROI, which accurately extract the features that we have in the image. I mean the ROI that we have. And it also avoids the misalignments that are caused by the quantization techniques that are applied in the ROI pooling layer too. So this helped us to segment each and every ROI that we had. So this is the general architecture of mask RCNN, and we'll be providing the results at the last. So this is just the architecture, and our proposed model is DeepLab V3. It employs a address convolutional layer because it has two types of techniques, like the pairwise technique and also the depthwise techniques, which helped us to capture the features that are present in a holistic way. And it uses the spatial pyramid pooling layer to enhance the contextual awareness too. So after building these two models on our data, this is how our performance will be. The mask RCN is getting overfitted, as we can clearly see the validation curve is going way below when compared to the training curve. And in deep lab v3 it is getting converged, so we can clearly see that the model is built in a proper way. And this is how the segmentations were like. And after that, we built one linear regression model, which helped us to map the number of white pixels that we have to the circularity index, so that for each and every cell will be mapping its number of pixels to its corresponding circularity index, so that if given a new image, will be segmenting each and every cell from that image, will be calculating the number of pixels in it to the circularity index, and will be predicting it, right? So that's how our pipeline works. And in conclusion, our work not only advances the understanding of plant cell morphology, but we can also clearly see that the advanced deep learning techniques that are being applied over here really gives better results in instant segmentation. So as we had only 28 images, we can just propose this pipeline, and it was really hard for us to avoid the model to get overfit on the data. So in future, we are trying to extend the model by using more number of images, so that it might perform way better when compared to these models right now. Thank you.